Morning, everybody, and good afternoon to those in foreign parts. This is Jeffrey G. I'm the NDF Executive Director. I've been aboard with the organization since January, and I'm delighted to meet you all. It's Sunday morning in Los Angeles, so you can see it's Sunday casual. It's a long weekend, Memorial Day, so we're all looking for good weather and some barbecues. I'm delighted to be able to introduce Dr. Kelly Crowe. Dr. Crowe is an assistant professor in the Department of Biology. She holds a Ph.D. in molecular, cellular, and developmental biology from the Ohio State University, as well as an MS in biology from Missouri State, and a BA in biology and psychology from Drury University. Her research interests include utilization of cell cultures in models to understand physiology and processes relevant to muscle diseases such as GNE. So Dr. Crowe will be giving her statements in a few minutes, and our moderator today is Dr. Noah Weisleder. Noah received his BS in biotechnology from Worcester. I think in England it's Worcester. I'm not sure in America whether it's something different. Polytechnic Institute, and a PhD in cell biology from Baylor College of Medicine. He conducts his postdoctoral studies at Robert Wood Johnson Medical School, where he joined the faculty as assistant professor in the Department of Physiology and Biophysics. Currently, Dr. Weisleder is professor and director of graduate studies in the Department of Physiology and Cell Biology at the Ohio State University, Wexner Medical Center. I'm now going to hand it over to Dr. Crowe. I thank everybody for attending. I see we have a nice group of participants, and of course the session is being recorded and will be available after the event. Dr. Weisleder will follow Dr. Crowe and be available in the Q&A, so I'll leave it to Dr. Crowe.
All right, and thank you all for taking the time to join us, especially on a Sunday morning, I guess Sunday evening for some of you. But I'm really excited to share with you some of the work that my lab has been doing over the past year. As Jeffrey mentioned, I'm an assistant professor at Mount St. Joseph University, which is in Cincinnati, Ohio. And so for the past 4 years, my lab has been working on GNE myopathy, and specifically looking at biomarkers for therapy in GNE myopathy. And so I'm going to share some of that work with you today. And so again, I am funded by the NDF to perform this work looking at biomarkers for gene therapy, and I'll tell you about some of the work that I've been doing and why I think that this biomarker work is so important for developing these therapies for GNE myopathy. So I won't belabor the point too much about the disease GNE myopathy, because I know that you all know very well about the characteristics of this disease, but just in very brief, GNE myopathy is rare disease leading to the decrease in muscle mass and strength, and it is caused by genetic mutations, and specifically genetic mutations in the GNE gene that affect the sugars that go on the cell membrane. And we'll talk a little bit in a second about what that looks like. On the left here, looking at some MRI work showing the changes in muscle mass over time over the progression of GNE myopathy. But let's take a look at what GNE myopathy looks like in the cellular sense. So GNE is a gene that makes a protein. And so GNE is specifically a gene, so a piece of DNA found here in the nucleus. So if this whole thing is a cell here in the nucleus, we have the DNA, which has the genetic codes. And part of that genetic code is this GNE gene. And so the GNE gene is going to be made into a protein that's called the GNE protein. Lots of different proteins are made from this DNA in the nucleus with a lot of different jobs. And the specific job of this GNE protein is to make a sugar. And the sugar we call sialic acid, which you'll see abbreviated as SA sometimes. So this GNE protein is part of a long pathway of different proteins that all work together to produce this kind of final product, the sugar sialic acid. And sialic acid from there is going to be sent out, and it's found capping these sugar chains outside of the cell. And so there are lots of different sugars that are kind of all in a line, and typically the very last sugar on the end of that sugar chain is going to be sialic acid. And so because of this placement of sialic acid, because it's right at the very end of all of these sugars, it plays a really important role in cell functioning. And because if you're kind of headed at the cell, the first thing you'll run into is that sialic acid here. And so its placement makes it very important as a sugar. So in the case of GNE myopathy, let's take a look at how this changes. So first of all, there are many different mutations, potential mutations in the GNE gene. But the result here, the GNE protein still gets made, but it's not going to have the same sequence that it would otherwise. So the protein is going to be mutated. And so the GNE protein is still able to do its job in working in that pathway to make the sugar sialic acid, but the mutation makes the protein less efficient at its job. And so many different mutations in the GNE gene lead to mutations in the protein that end up producing less sialic acid. And so then if you think about the fact that sialic acid kind of makes its way out and caps sugar chains typically, in the case of GNE myopathy, you're going to end up with some sugar chains that have sialic acid on them, but you're going to end up with these other sugar chains that are missing that sialic acid cap. And so we call these acylated or without sialic acid. And so you end up with these sugar chains that don't have sialic acid on them because of this original GNE mutation. And so I will add that the pathophysiology, kind of the mechanism of why the decrease in sialic acid and why the GNE mutation leads to muscle disease specifically, we're still working very hard on that. We don't have a complete picture of the mechanism. And so if you're thinking that I'm not explaining why a GNE mutation specifically causes muscle disease, it's still something that we're working out. But what we do know, thanks to some fantastic work from my colleagues in the field, is that you do end up with less sialic acid in many cases as a result of mutated GNE genes. And I will add that depending on the specific GNE mutation, there may be more or less of a deficit in the sialic acid. So you might have a more or less striking decrease in the amount of sialic acid. But one therapy that is that's very promising for GNE myopathy, and in fact that the NDF is working very hard in, is gene therapy. And gene therapy is exploding as a field, and for good reason, because gene therapy is able to use a gene itself, to use DNA in order to treat a disease. And so let's take a look at what that looks like for GNE myopathy. So again, as I mentioned, in the case of GNE myopathy, you start off with a mutation in the GNE gene, and less sialic acid is produced. And so you have a decrease in sialic acid on the surface of the cell. With gene therapy, you are introducing the gene back into the cell. So in this case, you would be introducing a GNE gene that's not mutated and putting that into the cell, into the nucleus, so that it can make the corrected GNE protein. And so you still would have that mutated GNE gene in there, but because you've also delivered the non-mutated gene, it's able to make the non-mutated protein. The GNE protein can work in part of that pathway, making sialic acid, and the sialic acid can then cap sugar chains. I mean, that's a very promising idea for changing the course of the disease. So in order to get the gene into the nucleus, that's no easy task. In fact, delivery into the cell is the most challenging part of gene therapy. So in order to deliver the GNE gene into the cell, we have to use something called vectors. Vector is a delivery vehicle here. And so one vector has come up as particularly promising for gene therapy, and that is a specific kind of virus. And so we mostly know viruses as as giving disease, right? But really, viruses are evolved and adapted to deliver their genetic material into cells. And in fact, with gene therapy, that's exactly what we want. We want to be able to deliver genetic material into cells. And so what we can do is engineer a virus so that instead of having all of the viral genes inside, we put in a copy of the gene we want to deliver. In this case, the GNE gene. And then the virus, we most commonly use one called adeno-associated virus because it's safe and effective at delivering. The virus will then take that gene, deliver it into the cell, and then the non-mutated copy of the gene can make the GNE protein again and deliver that sialic acid out to the cell surface like it's supposed to. So we're very excited about adeno-associated virus, or AAV, to deliver the GNE gene into cells. And the reason why we're so excited about AAV in particular is because it's been really promising in other neuromuscular diseases. And so you may have heard of Duchenne muscular dystrophy, some really fantastic work on AAV-based gene therapy there. And then also I'll highlight here spinal muscular atrophy, or SMA, and another neuromuscular disease that has had really fantastic promise with AAV-based gene therapy. And in fact, this is the kind of final product for AAV gene therapy for spinal muscular atrophy, Zolgensma. And so the promise of AAV in these other diseases makes us as GNE myopathy researchers really excited about the development of GNE gene therapy for GNE myopathy. And like I said, the field is really just exploding. Like I told you, with gene therapy, the trick is being able to actually deliver the gene into the cell. That's the trickiest part. And so because of that, we need to have a way to figure out kind of experimentally how many cells have been effectively, we call it transduced, but have effectively gotten the gene therapy inside of them. And that's really important because once you give a dose of a gene therapy, you need to know kind of how effective that is at getting into cells. So in the context of a clinical trial, if you see a clinical effect, you want to say, okay, how much of that gene actually had to get into cells in order to see this clinically meaningful effect? And that helps you to say, okay, in future doses, how much do we need to give? Do we need to give more in order to see a better effect, or is the dose that we gave okay? And it also helps to show the FDA that the clinical change that you're seeing, whether it's preservation of muscle strength or preservation of muscle mass, it helps the FDA to say, hey, this is a real effect if you can actually see how many cells have gotten the gene therapy. So being able to visualize which cells have gotten the gene therapy is really important. And so I just want to show you what this might look like over here on the right-hand side. And so this is work from Paul Martin's lab at Nationwide Children's Hospital. And this is a gene therapy that Paul's lab developed for a different muscle disease, Duchenne muscular dystrophy. But it does help you to see what it might look like to visualize how gene therapy has gotten into cells. So here you can see all these numbers, and all these numbers are different doses of the gene therapy that have been given to a mouse. And then you're kind of looking for these green circles that start appearing, because here we're looking at a slice of skeletal muscle. So the gene therapy is being delivered to skeletal muscle, and each time you can see this little green glowing circle. That means that each circle is one muscle cell. And when it's glowing green like that, it means that the gene therapy has effectively gotten into that cell. And so you're able to visualize it using a biomarker. And so you can see that as the dose increases, as these numbers get higher, you can see that more and more cells are glowing green, meaning that a larger percentage of the muscle cells have effectively gotten that gene therapy inside of them. So in order to do this, in order to be able to visualize how many cells have gotten a gene therapy, like I said, you need a biomarker. And for GNE myopathy, my lab is really excited about using something called lectins in order to visualize which muscle cells have been treated by a gene therapy. So put very simply, a lectin is a sugar binding protein. There are a ton of different lectins, usually isolated from plants, that can be used to bind sugars or carbohydrates. And the particular lectins that I'm excited about are those that can be used to detect sialic acid. As you remember, sialic acid is decreased after GNE mutation. And then once gene therapy is delivered, that sialic acid level should be restored. And so lectins could be used to detect those changes in sialic acid that you would see, that restoration of sialic acid as gene therapy gets into a particular muscle cell. So really excited about using lectins as biomarkers for GNE myopathy gene therapy. There are two types of lectins that my lab uses. So some lectins actually bind sialic acid themselves. So remember, sialic acid is usually capping this long sugar chain. And so some lectins, which I'm calling lectin A here, would directly bind sialic acid when it's present. And so these lectins have attached to them something that's green and glowing. And so that way you can see how many lectins are actually bound to sialic acid. Other lectins, though, don't bind sialic acid itself, but they bind those sugar structures that are underneath sialic acid. So when the sialic acid is missing, then lectin, the second class of lectin, which I'm calling lectin B here, would be able to bind and bring its glowing tag with it. And so I can use both of these types of lectins to measure either the presence or the absence of sialic acid. Let's take a look at what this might look like in GNE myopathy. So in the case of these sialic acid binding lectins, you can see here, this is a non-GNE myopathy muscle biopsy. And you can see each circle here, just like we showed before, each circle is a single muscle cell. And this glowing green that you're seeing means that you're able to visualize the sialic acid that's surrounding each individual muscle cell. Over here, this is looking at muscle of a patient with GNE myopathy. You can see that there's a decrease in this glowing. And that's because when there's reduced sialic acid, in the case of GNE myopathy, this lectin is less able to bind. And because the lectin has that glowing tag on it, you can see that the reduced sialic acid means that there's a reduction in the green glowing surrounding the muscle cell. And the same kind of principle applies for the second class of lectin, which binds the underlying sugar structures. So this lectin binds those sugars that are underlying sialic acid. So in the case of non-GNE myopathy muscle biopsy, you can see that there isn't very much green glowing here. And that's because most of these sugar chains are capped with sialic acid. And so lectin B, the second class of lectin, isn't able to bind. It's being blocked out by sialic acid. But in the case of GNE myopathy, where there's this decrease in sialic acid, lectin B is able to bind these underlying sugar structures, and you can start to see this green glowing surrounding each muscle cell. And in this case, you're saying, hey, there's not sialic acid here. And so you can see how these lectins can reflect either the presence or the absence of sialic acid, and you can visualize that on a cell-by-cell basis in the muscle. And that's exactly what we're looking for a GNE myopathy gene therapy biomarker. Okay, so what my lab is interested in is that I showed here lectin A and lectin B as these two different classes, but it's actually a lot more complex than that. There are a ton of different lectins in each of those categories. So some of those lectins directly bind sialic acid itself. And so we would expect to see those decreased in GNE myopathy, but these are all different specific subtypes of lectins. And they recognize slightly different contexts of sialic acid. Like one might recognize sialic acid only if it's bound in a very specific direction to the sugar beneath it, or a slightly different sugar beneath it. And so while all of these lectins here bind sialic acid, they might show different abilities to reflect changes in GNE myopathy based on their specificity of sugar binding. And so these ones down here, these three lectins recognize those underlying sugar structures. And again, there are three different types of lectins that recognize those underlying sugar structures. And so all of these have been shown in these different contexts to recognize sialic acid changes in GNE myopathy. But my lab's question is, which of these lectins would be the most promising, would show the biggest difference in the case of GNE myopathy and in the largest number of different contexts? So which one of these would be the best as a biomarker for GNE myopathy gene therapy? And so what my lab does is look at which of these lectins that all show changes in the case of GNE myopathy, which should be the most promising as a biomarker, which has the largest change that you can visualize and is the most consistent in its binding. And so because of that, which would be the best to visualize that GNE myopathy gene therapy? So in order to test this, my lab takes the approach of using a lot of different models and changing sialic acid and measuring the ability of each lectin to reflect those sialic acid changes. And so sometimes, I'll say that we use a lot of different models because no model is going to be exactly perfect in mimicking a patient muscle cell that has received a gene therapy. So we use different models to kind of mimic what that would look like, and each gives us a little piece of the puzzle and allows us to predict which would be most effective as a biomarker. So one thing that we use is cells in a petri dish. So you can grow up cells and keep them alive outside the body in a kind of a warm, moist incubator. So we use those. We use biopsies from GNE myopathy patients as another very critical piece of the puzzle to understand these biomarkers. We use a zebrafish model that's developed by Dr. Landaya and Dr. Stella Matrani-Rosenbaum in Israel, really fantastic work by that group. And then we also use a mouse model of GNE myopathy to look at what that would look like in a mouse model. And so each of these methods, while they're very different, give another important vantage point, another different piece of the puzzle. And so we can say, hey, of all these lectins, which is the most robust, which shows the biggest difference in all of these contexts and would be most useful as a biomarker? And I want to show you just one more thing here. So you'll see in my data that sometimes we're looking at what happens when GNE is mutated. And so that would be the condition we looked at before, mutated GNE gene, making mutated GNE protein, and less sialic acid is produced overall. But sometimes we also use an experimental model using an enzyme called neuraminidase. And so in this case, you also have decreased sialic acid, but the reason is different. Neuraminidase is an enzyme that chops sialic acid off. And so in this case, the GNE gene is unmutated. The protein is still producing plenty of sialic acid, but neuraminidase is an enzyme that's kind of chopping that sialic acid and removing it from the sugar chains. And so we use both of these approaches to model that decrease in sialic acid that you would see in the case of GNE myopathy. And so you'll see in my data slides, in the bottom corner, I'll have one of these tags to show whether we're looking at cells in a Petri dish, muscle, this zebrafish model, or mouse. All right, so starting off here, we are looking at cells, and these are HAP1 cells that were really graciously provided by the Lec lab at Yale. And so these are, here we have some cells that are kind of standard cells, nothing has happened to them. And then we have a couple of different conditions. These are cells that have been treated by neuraminidase to kind of chop off the sialic acids. And these are cells that have been genetically mutated to mutate the GNE gene. And here we're looking at a lectin called VVA. And so because it's binding those underlying sugar structures, you would expect to see an increase in staining in the case of neuraminidase treatment, or in the case of GNE knockout. But we can see that VVA in this context, in these cells, isn't showing a very striking difference in these different conditions. And so with VVA, we did not see a striking difference in the amount of green glowing that we're seeing after sialic acid is removed. However, when we look at a different lectin, this one is called SBA. It's again one of those lectins that you would expect to be increased when sialic acid is removed because it's binding those underlying sugar structures. In this case, SBA did show a really striking change when either sialic acid is removed with that enzyme, neuraminidase, or when sialic acid has reduced production. And so in this case, when we're looking at these cells in a Petri dish, SBA looked really promising in reflecting that decrease in sialic acid that we're seeing as a result of the removal or reducing the production. Another lectin called PNA, again binding these underlying sugar structures, also looked really promising in these cells in a Petri dish. So you can see again that as sialic acid is either removed or not produced, we're seeing a really striking increase in that green glowing, in that fluorescence, indicating that this PNA lectin is really robust in this model in cells in detecting the differences when sialic acid is present or absent. But we wanted to look at these lectins in human muscle biopsies as well, either from non-GNEM patients or GNEM patients. And so I'll show a few lectins in the context of these muscle biopsies. And so whereas before we're kind of seeing cells that are growing in a Petri dish, now we're back to seeing our little green circles. And remember, because you're looking at a slice of muscle, each little green circle is a single muscle cell. So again, back to VVA, one of those lectins binding the underlying sugar structures, we tested that in these muscle biopsies, and we didn't see a strong difference here. And so when we quantified this, here we have non-GNEM on the left and GNEM on the right, and we're basically looking at how much fluorescence there is, how much it's glowing. And we didn't see a difference between non-GNEM and GNEM patient muscle. However, when we looked at SBA, that second lectin from before, and we started to see more of a difference. So in the case of a non-GNEM patient muscle biopsy, there's not much green glowing that you can see here. But in the case of GNEM, you can start to see those muscle fibers being circled. And in fact, when we quantified it, again, with that same method, comparing fluorescence, we saw an increase in the in the fluorescence with SBA, and this was a statistically significant increase, meaning that it's meaningful there. And this work was done in a collaboration with the Nishino lab in Japan, as well as the Weissluder lab, Noah's lab at Ohio State. And so we have a fantastic network that facilitated this work. But you can see that there's kind of an increase in fluorescence, meaning that SBA was able to show those changes in fluorescence as a result of sialic acid being decreased. And then finally, we looked at PNA Lectin again in the case of non-GNEM muscle here versus a muscle biopsy from GNEM patient. You can see that PNA is strongly surrounding the muscle fibers after kind of in the case of the GNEM patient muscle biopsy. And again, when we quantified, we saw a statistically significant, meaningful increase in the fluorescence. And so PNA seems to be, in the context of both cells and in the context of patient muscle biopsies, seems to be really promising in the ability to reflect the changes in sialic acid that you see in GNE myopathy. And next, I want to show you just a couple of things that we're starting to work on with zebrafish and then with mouse models. Before we start looking at the data here, so everything I've shown you so far is in human cells. So either human cells in a petri dish or human muscle biopsies. But it's also really important when developing a biomarker to not only look in humans, but also to look at animal models of the disease. And so the reason why that is because as gene therapy is being developed, it's also being tested before it can ever get to clinical trials. It's tested in animal models, such as a mouse model. And so when you're looking at a biomarker to say, which cells has my gene therapy gotten into, it's fantastic and critically important to know if that's the case in human cells, but it's also really important to know, hey, when I'm testing this gene therapy in an animal model, does my biomarker work the same way? Is that same biomarker able to show changes in the mouse model? And that's really important for GNE gene therapy development. And so that's why we're looking at these lectins in animal models. And so I'll show you some of the early work that we've done this year. So the zebrafish work done in collaboration with Dr. Alandaya in Israel. So here, their lab has developed a model in which the GNE gene is either one copy is knocked out or both copies are knocked out. And so we're still working on optimizing this model here, but what I want to show you is that as you are looking at, again, a cross-section of muscle, we were very excited that we are able to see and visualize muscle fibers in this zebrafish. And so it's a little harder to see here because these are zebrafish larvae and they're very, very small. But you can see these little green circles, particularly right here. And these green circles are these muscle cells, and we are able to use our lectins to visualize sialic acid in the muscle of this zebrafish model. And so as we're continuing to optimize and develop this lectin staining, we're really excited to see if these lectins are able to reflect sialic acid changes that would happen when the GNE gene is knocked out in this zebrafish model of GNE myopathy. And we're also interested in mouse muscle because mouse is a critically important model for gene therapy development. And so to show you some of the early work that we've done here, again, we are looking at muscle cells. Each one of these circles is a muscle cell. And so let's focus in here on the left with the PNA lectin. Again, PNA Lectin binds those underlying sugar structures, so you would expect it to glow brighter when sialic acid is removed. And so here we have taken what we call wild type, or just kind of non-mutated, so GNE is non-mutated mouse muscle, and we have treated it with neuraminidase, which removes that sialic acid. And we have been able to see the change in sialic acid being reflected by our PNA lectin. And so PNA more strongly fluoresces when that sialic acid is removed, indicating that PNA would be able to detect those changes that you would see in mouse muscle when sialic acid levels are changed. And so we're very excited to use PNA as well as some of these other lectins that we're looking at, such as MAA, in mouse models of GNE myopathy, to look at their ability to reflect the extent of sialic acid loss in GNE myopathy, and then be able to look at gene therapy when that sialic acid has been restored, and see if our lectins are able to go the reverse direction, are able to revert back to looking like this when the sialic acid is restored by GNE gene therapy. So overall, what I've been showing you today is that when you use that lectin staining, you are able to see the changes in sialic acid that happen in the case of GNE myopathy, and when sialic acid levels are decreased, and also in the case of GNE gene therapy, mimicking GNE gene therapy, and when those levels would be restored. And so if this lectin staining can show the presence and absence of sialic acid, then it could be used to measure that gene therapy and measure those sialic acid changes that would occur. And as I told you, we're using a lot of different models, and we're looking for which lectins are the most robust change, the most reproducibly in all these different contexts. And so far, we show that PNA is particularly promising in a lot of these contexts. We saw PNA having these really robust changes in fluorescence in cells. We saw it in the human patient biopsies, and then we also saw it in the mouse model, the mouse muscle that had been treated with that neuraminidase enzyme. And so PNA seems to be very promising in showing and reflecting those sialic acid changes. And as we're continuing this work, as I briefly mentioned, one thing that we're interested in doing is using that lectin panel in the available mouse models of GNE myopathy. So we showed it with neuraminidase, where that sialic acid is removed by an enzyme after the fact. But we also want to look at the available mouse models to see if we are able to visualize the changes in sialic acid there. And then very critically, and we're also working with many of my fantastic colleagues and collaborators at the NDF to test our lectin panel in the case of GNE gene therapy preclinical trials. So when that GNE gene containing virus is delivered into mouse muscle cells, if we are able to use our lectins to visualize the changes in sialic acid that would occur after that. And so those are the next steps that my lab is actively working towards. But overall, finding a biomarker that allows you to visualize which cells have gotten the gene therapy is very important, and we're excited about lectins. And we're excited about validating our lectins in both human cells and also in animal models, so that we can know that we can use our biomarker for both preclinical and clinical trials for a GNE myopathy gene therapy. And of course, I want to thank the fantastic folks in my lab who have actually done this work, notably Christy Sattler, Megan Gilpin, Kara Bradley, Mackenzie Roth, and Meg Pointer, who have done the work that I've shown you today. I'd like to thank all of my fantastic collaborators who have provided samples and shared protocols and really allowed all of this work to occur. The LEC lab, Yale, the Nishino lab in Japan, the Weiss-Lueder lab at Ohio State, the Matrani Rosenbaum lab at Hadassah, Paul Martin's lab at Nationwide, all very fantastic researchers that I'm very excited to work with. And then I would also like to thank all of you, the patient community, because you are the reason why we're doing this work, and we are very grateful for your continued interest and support, because we believe that this work is very critically important, and we're excited to continue it and excited to continue this biomarker development. And of course, thank you to the NDF for not only funding this work, but for putting together this speaker series so that my lab can share our work with all of you. And we're very, very excited to do so. All right. Well, thank you very much.
Thank you very much, Dr. Crowe. That was a great lecture, and we actually have a couple of questions in the chat already that I'd like to ask you your opinion about. The first one is, are there any significant differences in how the lectins perform in detecting changes in sialic acid based on the different variants of GNE mutations?
Yeah, that's a really fantastic question. So the lectins would be expected to work more robustly with some GNE mutations as compared to others. And that's something we're very actively interested in, because what previous data has shown is that some GNE mutations cause only a more slight reduction in the sialic acid levels, while other GNE mutations cause a very robust decrease. And so this is something that we're actually looking to assess very, very soon, because I told you that we're looking at different mouse models of GNE myopathy. And so one mouse model has a mutation that is expected to produce a very slight deficit in sialic acid, and another is expected to have a very significant deficit in sialic acid. So as we move towards testing our lectins in these different mouse models, I'm excited to start to address that question of the lectin's ability to show these changes in the case of different GNE mutations, because you're absolutely right that it's critical to have a biomarker that can be used for different patient populations, because there's this a significant mix of different mutations in the GNE myopathy patient community. Yeah, great question.
Thank you. And so a second question here is, in muscle biopsies, how much variation do you see amongst muscle fibers, especially since biopsies can be from a less damaged section of the muscle in some cases?
Yeah, absolutely. That's another great question. And so in the case of the muscle biopsies, of course, as we know, muscle biopsies can show changes depending on where exactly that biopsy is gotten from, which muscle is being biopsied, and it's by no means a perfect measure. And so we do see some differences. So it's not every single GNE myopathy patient biopsy versus non-GNE myopathy patient biopsy looks the same. However, when we're able to test a larger number of these biopsies and then run statistics, we were able to see a statistically significant decrease. And actually, I might just pop back to that slide, if you don't mind. There we go. So for example, with this PNA Lectin here, so these are the values of the fluorescence, right? The fluorescent units of the non-GNEM patient biopsies. And over here, this is the fluorescence level of the GNEM patient biopsies. And you can see that there's hardly any overlap between the two. And so while there is some difference, so some of these GNE myopathy patient biopsies show more fluorescence and some show a little less, we're still able to see that in this case, it almost completely separated out between the two. And so this is hopeful for us that PNA is able to reflect the differences between non-GNEM and GNEM patient biopsies, even if not every GNEM patient biopsy is going to look the exact same.
Great. So if there's any additional questions, please feel free to put them in the question and answer panel. While we're seeing if any additional questions or follow-ups are coming along, I actually had a quick question I wanted to ask. And that is, you rightfully talked quite a bit about the impact that developing these biomarkers would have in validating gene therapy approaches for GNE myopathy. But in a broader sense, couldn't these biomarkers be used for any therapeutic approach targeting GNE myopathy?
Yeah, absolutely. So a therapy that is going to restore sialic acid to the cell membrane would cause a change in this biomarker. And that's really exciting because there is strong potential in the future that in addition to a gene therapy, a patient would also be treated with other types of therapies. And this is common in a lot of neuromuscular diseases, that as therapies are developed, multiple different therapies could be used on the same patient. And so this biomarker development is really exciting, as you pointed out, because it would be able to reflect the sum of sialic acid changes as a result of different therapies that could occur at the same time.
Thank you. Do you think we have enough versions of lectins? I mean, there's so many different lectins. These are all, for the most part, naturally derived products. And there's a limited number that are available at any time through suppliers and such. But do you think we have enough? Do you think you have optimized the level that's, or that your panel is sufficient? Do you think you would need more lectins to be able to have a broad-based representation of what the available probes are out there?
Yeah, that's a really fantastic question. So when I first started developing the panel that I showed at the beginning of the talk with all of the different lectins that I routinely test, I looked through the papers that all of my colleagues in the field have published. And so I looked for any lectin that has shown changes in the past. And so it's kind of taking all of these lectins that have shown changes in different contexts and comparing them head to head in these different model systems. But science like ours, kind of translational science that's looking towards therapies, is always built on basic science. And so my hope is that as basic science continues and potentially additional lectins are identified that bind sialic acid, I would be very excited to include those in my panel, because what my work is saying is that, you know, this lectin in my lab is the most robust, shows the most striking differences of the ones that we know about so far. And so if additional lectins are isolated and identified, then I would be excited to compare those to the lectins that we already have and say, is this better than the best ones we have so far?
Excellent. And we have another question in the Q&A section. First, thanking you, Dr. Crowe, for a very informative presentation. But the question is, out of the 4 different subjects used, namely cells growing in a petri dish, muscle fibers, zebrafish, and other mouse models, is there a particular mix of subjects that's critical for FDA and preclinical trial approval?
Yeah, that's a really fantastic question. So as I'm working together with my different colleagues in the NDF, the exact mix of what we would put in an application for the FDA is going to depend on a lot of different factors. And so I can't speak to what we will put in that FDA application, because I know that all of us are working towards the best application that we can put forward. So historically, different applications to the FDA, some have included these cell models, some have included, many have included those mouse models. And so I do think all of these different methods are pieces to the puzzle. And so as we start to put those together, then that's when we can have a very strong application. And so I don't know exactly what we will put together at the end of the day to make that strongest application. I do think that it's important to test all of these different pieces of the puzzle to converge on what we think the strongest data would be for that application. Yeah, really fantastic question.
Indeed. There's actually another one in the chat here. Are there any ways to amplify the signal from lectins?
Yeah, that's a great question. So when we are working with these lectins, the piece that I'm showing you is kind of the end of the day piece, kind of the final piece of the puzzle. But a lot of what my lab does is actually earlier than that. And so we are optimizing all of the different technical steps in order to get the best signal. And so the data that you're seeing is when we say, hey, if we use this detergent versus that detergent at this one step of the staining process, which gives us the most robust signal? And so that's a great question, because that's a really important part of what my lab does that I gloss over, is that highly technical troubleshooting to get the most robust signal that we can. And when we're very excited that in many cases after that really technical troubleshooting, we're able to see really striking differences. So when you think back to those pictures that I showed, in some cases it's a very dark picture and then a lot of green. And so those lectins are able to really show those differences in a very striking way. And it's all that nitpicky troubleshooting of the process that allows us to get those large differences. So yeah, fantastic question.
Excellent. I don't see any more questions in the chat box, or, oh, we have one more in the, just appeared. The question is, how far are we how far are we from starting a gene therapy trial in humans? 1 to 2 years, or 5 to 10 years, or more?
Yeah, and that's a really fantastic question. And unfortunately, one that's very challenging to answer. And so my piece of the NDF puzzle is really focused on this biomarker. And looking at, once we kind of have this gene therapy, how can we measure it? How can we view it? And how can we assess it? And so I know that a lot of my colleagues are working on the therapeutic development piece. And so I don't know that I have the best answer for that really important question. But I do know that my colleagues who are really focused on this have 2 priorities in mind. First of all, making something that is, getting something together that is safe and will work. And then secondly, doing that as quickly as possible. And so science isn't always a straight line. But I do know that the NDF and all of the fantastic scientists who are working on this gene therapy are looking to get something forward as quickly as possible that is safe and will work.
Exactly. And yeah, thank you. That's an excellent point. And also, I emphasize that we will have more people in the speaker series going forward who are working directly on development of gene therapies sponsored by the NDF as well, and they will probably have some more insight into that question as well. So we look forward to that and the future sessions here. But for today, let's thank Dr. Crowe for a great lecture and staying on to answer these really insightful questions. So we appreciate that. And in addition, I'd like to emphasize that our next session, today we had one of our really great early career investigators who's working with the NDF. But next time we have a very established investigator in the field of GNE myopathy research, Dr. Stella Mitrani-Rosenbaum from Hadassah University in Israel, who has made many seminal contributions to this field. And really, it should be a great seminar next time about the current state of the field and some of the work taking place in her laboratory that's helping to accelerate development of therapies for GNE myopathy, both directly and also by working on developing new models that can be used to test these therapies. So that session will be on Sunday, June 26th at 9 a.m. Pacific time, 12 noon on the East Coast, and at the appropriate times throughout the world. So we appreciate you all being here today and hope you have an excellent rest of your weekend, and we will see you at the next session. Jeffrey, did you have anything you wanted to add?
Yes, I just wanted to thank everybody for their participation, our speakers in particular. As you can see, the NDF is really moving forward in all aspects to find solutions. And I think Dr. Crowe's speech today really emphasizes the work we're doing. I also would like to thank everybody for their participation as patients, as scientists, and as volunteers in every sense. And I also hope that everybody has a wonderful long weekend. Thank you.